So in this video, I'm going to explain the fundamentals of protein structure. So proteins are obviously the most important molecules in the cell in many ways because they produce all sorts of functions. So proteins, they can be enzymes, they can be receptors, they can be transporters, they can be transcription factors, they can be all sorts of different, have all sorts of different functions within the cell. And their function is going to be linked to their underlying structure. So you need to understand their structure in order to understand how you might function as a receptor, for example. Okay, so there are 4 levels of protein structure that we need to consider. So the first one is the primary structure. Okay, so the primary structure is the sequence of amino acids. Okay, so inside the cell we will have a bunch of amino acids that are just free inside the cell. So I'm just going to have a couple of them there. So each of these different colours is a different type of amino acid. Okay, so these are going to start free inside the cell. Okay, so we've got, and there's actually 20 different types of amino acid. I've just shown 4 for there. Okay. So the primary structure is the process of turning those into a chain of amino acids. I'll just quickly do this here. Okay, so we've got a chain of amino acids that is obviously produced during the process of translation. Okay, so this is catalyzed by the ribosome. So there we've got our amino acids, and they are linked there with peptide bonds, which, as I say, are formed during translation. Okay, so the peptide bond forms in this way. So we've got H2N, CH, there's an R group, COOH plus. There's another one, H2N, C, COOH, H, and there's the second amino acid. So in this case, this would be the yellow amino acid and the green amino acid. Obviously, we could substitute in some real names there. So this is the first one. When they come together, we lose a molecule of water and we form a bond that is, so we've got H2N, C, H, there's the R group, then we've got C, double bond O, N, H, C, and there's the R group sticking on there. Okay, so again, that is the orange one, that's the green one, okay, from my chain over here, and what we formed in the middle is the peptide bond. There's another video for you about the different amino acids and all of their properties, so I'm not going to sort of go through that now. There's another video for you to have a look at. Okay, but the primary structure is all about making that sequence of amino acids, and it's held together. The most important thing we need to know is these are peptide bonds, which are proper covalent bonds. They're covalent bonds that form between the carboxylic acid group and the amino group to create this characteristic peptide bond structure. So that's the first one. Obviously, the more amino acids you add to the chain, the more peptide bonds you have. And per protein, and it's really variable, but you're roughly looking at about 1,000 amino acids in a protein as a good basic step. But some of them are a bit smaller than that, some of them are a bit bigger than that. But roughly, we've got a chain of about 1,000 amino acids. Okay? So that's primary structure. Let's think about secondary structure. Is regular structural motifs or patterns, I guess. Okay. So secondary structure is all about hydrogen bonds from the backbone of the protein. So let me just explain what I mean by that. Okay. So here we've got our sequence of amino acids in a straight chain. Well, proteins aren't straight chains, they're complicated things. Okay. So what we have is characteristic ways that these can form higher level structures. But they're underpinned by a particular pattern of hydrogen bonding. So it's not random bonding, it's a very precise sort of bonding that we're thinking about. So if we've got an amino acid here, okay, that has formed a peptide bond. Okay, let's do the rest of it dotted there. Okay, so we've got our peptide bond in the middle there. Okay. If there is another peptide bond somewhere near, then we can start to form other bonds. Just write that out. Okay, so here we've got two peptide bonds, okay, and because of the biochemistry of that, we've got some differences in electronegativity. So oxygen we know is a little bit electronegative, nitrogen we know is a little bit electronegative, which makes that a little bit electropositive. So there's an attraction that can form between the peptide bond of one chain and a peptide bond in another chain. Okay, so that we call a hydrogen bond. So it's not a covalent bond, it's a weaker bond than that, and it's due to the formation of these bonds from the backbone. Okay. And you probably know that there are two types of secondary structure that we see again and again and again. So we have the alpha helix. Okay, so the way that an alpha helix works is like this. So we've got, there's our helical structure, okay, and we've got these hydrogen bonds forming between stages on the spiral, effectively. So there'll be a hydrogen bond in there, there'll be a hydrogen bond in there, there'll be a hydrogen bond in there, hydrogen bond in there. Okay, so remember these are forming from the backbone, from the peptide bonds. Okay, so in an alpha helix, notice that the R groups are not involved in this at all yet. Okay, so if I just have my R groups, I'm just going to do all of them in orange for the moment. Okay, the R groups are not involved in these bonds. The R groups are sticking out the side of the alpha helix like this. Okay, they're sticking out the side, they're not doing anything chemically interesting yet. Okay, this is all about hydrogen bonds from those peptide bonds in the backbone. The other secondary structure that you can have is the beta sheet. So the alpha helix is within one piece of protein. With the beta sheet, what you have you need two bits of protein to come together. So if we've got there, we've got the O, and there I've got the NH. Then we've got the bond forming between them. Okay, and that will happen again down the protein. Okay, so again, it's still hydrogen bonds from the backbone. So you're asking where are the R groups? The R groups are sticking in and out of the page here. So here's an R group. There's one sticking back there. There'll be an R group sticking out here. There'll be an R group sticking there. Okay, so they're kind of above and beyond, below the plane of what we're looking at. Okay, so there'll be a C double bond O in there, for example. Okay, beta sheets. We've got two different versions of them. So you can either have what we call a parallel beta sheet. So these strands have a directionality to them. So we can either have the two sheets are going in the same direction, and we've got hydrogen bonds between them. Okay, so that would be an example of a parallel beta sheet, or we could have an anti-parallel, which is actually more usual. hydrogen bonds forming between there, like that. So in our antiparallel, we might actually have it be like that. So this is where we're starting to get higher order structures in the protein with these sheets. Okay. You might be asking, why do we only ever see alpha helixes and beta sheets? The reason that we only see these is because they have maximal internal hydrogen bonding. So what that means is that every single peptide bond is hydrogen bonded to another peptide bond. There aren't any free groups kind of hanging around. Every single peptide bond is bound to another one. Okay, I'm just going to highlight their peptide bonds on there. That might be helpful. Okay, every single peptide bond is bound to another. So here we'd also have a peptide bond going off in that direction. We'd have one going off in that direction. And that means that we exclude water from the structure. So with protein structure, water always wants to come in and disrupt it. Water wants to get in, wants to bind to stuff, and that will sort of tear the protein apart. So alpha helices and beta sheets are really good because every single peptide bond is bound to another one, so there's no space for any water, so the protein's nice and strong. Okay. So what do we mean by tertiary structure? So tertiary structure is the unique three-dimensional structure, and that is formed with bonds with the R groups. Okay, so notice the R groups haven't done anything up to now. For primary and for secondary, they've just been sitting there sticking out to the side. It's the tertiary structure where the R groups get involved. So let's imagine we've got a bit of alpha helix here with an R group, some R groups sticking out. Okay, and then I've got in my protein, I've got another bit of alpha helix with some other R groups sticking out. Okay, those R groups can bind with each other. Okay, so these two R groups here might be attracted to each other and be able to form a chemical bond. Not all R groups can, but most of them can. So what sorts of bonds are we thinking about? Well, what we could be thinking about is a hydrogen bond. There are hydrogen bonds in the three-dimensional structure. So if the R group has a chemical group in it that allows you to form a hydrogen bond, so let's say it's got an OH group in it, we know that OH groups are polar, so they can form hydrogen bonds. So if if both R groups have a polar component, so if both have got something polar in there, they might be able to form the hydrogen bond. You might be able to form an ionic bond, which is where you've got something positive and is attracted to something negative. Okay, so if you look at the amino acid video, you'll see that there are amino acids that have got positive and have got negative bonds. So a lysine, for example, which is positively charged, might be attracted to an aspartate, which is negatively charged. So if those two amino acids are there, they'll form an ionic bond. You can also have disulfide bonds, and they only form if you've got two cysteines next to each other. Okay, so we've got two cysteines next to each other. The sulfur in there in those two can react with each other, and you form what's called a disulfide bridge. That's a really strong bond. But what you can also have are effectively hydrophobic interactions. So if you've got two hydrophobic, so two non-polar groups next to each other, so let's say you've got an alanine and phenylalanine, for example, they're both made of carbons and hydrogens. They won't be able to form any bonds with each other, any sort of formal bonds, but because they're both non-polar, they will be attracted to each other because, again, we're trying to exclude the water. Okay, so depending on what R groups you've got, it depends on which sorts of bonds you might be able to form, but we've got those different types of bonds. Okay, so the three-dimensional structure of a protein, you'll then have bits of alpha helix, bits of beta sheet that are then attached to each other through interactions with the R groups. Okay. Last bit is quaternary structure. Which is all about interactions, bonds between proteins. Okay, so up to now, everything has been in with a single chain. So we had our 1,000 amino acids, they had their primary sequence, got the secondary structure, got the tertiary structure. All of that is within one protein. So a single chain of amino acids obviously has primary structure, can form secondary structure, can form tertiary structure. To have quaternary structure, you have to have 2 or more proteins there. So 2. So what we might have here, I'm just going to do a very quick cartoon of this because I'm running out of space. So here's a protein. Here's protein A. Here's protein B. Okay, so we've got 2 different proteins that obviously have R groups sticking out on the surface. Okay, so again, it's R groups are responsible. Okay, and again, we can have bonding between R groups. This one, they were inside the same protein. Quaternary structure, they've got R groups that are pairing between 2 proteins. Okay, so again, we can have different types of bonds. So we can have ionics, we can have hydrogen bonds, we can have hydrophobic interactions. We tend not to get disulfide bonds between 2 proteins. That's more unusual. But the others we can have. So we might have 2 proteins that both have got actually quite a hydrophobic bit on the outside. That would be non-favourable to interact with water. But if those 2 hydrophobic bits can stick to each other, so let's imagine so all of the R groups here, and all of them are here, if they're all hydrophobic or non-polar, then they might be able to stick to each other. Okay, so A could stick to B because all of the R groups at the interaction site share the chemical property of being hydrophobic, so effectively they stick together. Okay, so quaternary structure is bonds between proteins. And this means that if we're thinking about the function of a protein, then this starts to set up us for that function. So let's imagine we've got a protein, so I'm just going to draw it here, just a cartoon of a protein, okay. And let's say that this needs to interact with a particular molecule. So I don't know, let's have a molecule. I'm just going to make one up. Yeah, let's have and let's have an OH there. And let's have an NH2 there. Okay. But this is all carbon and hydrogen. So if here's the protein, and this might be a signalling molecule, it might be a substrate for an enzyme, might be something it needs to transport, doesn't really matter, okay, for this example. Okay, the protein will have R groups over the surface. Okay, inside the protein will have been the secondary structure, so there might be a bit of alpha helix there, a bit of alpha helix there, bit of beta sheet in there, bit of beta sheet in there. Okay, so that's the secondary structure. Okay, they're then arranged in this unique 3-dimensional arrangement because they've got internal bonding. So we've got some bonding in there, we've got bonding there, bonding there. Okay, so that is tertiary structure holding the whole thing together. Okay. And then the R groups that are pointing out on the surface might affect how this protein interacts with a substrate. Okay, so for example, this R group here, now let's start with these R groups. These R groups here, I would expect to be non-polar. because this molecule is relatively non-polar in this side. It's carbons and hydrogens. There's no interesting chemistry on here. So I would expect these groups to be non-polar, and they're held together because of the unique protein structure. This R group here is next to an OH group, so I would expect that to be a polar one, so that it could form a hydrogen bond with that OH group. And then this R group here, well, we probably know that NH2 when we're in the cell is nearly always NH3+. So that's positively charged. So I would expect that this R group here would be negatively charged. So that might be something like aspartate. Okay. So polar amino acid, that might be serine, for example. That one might be something negatively charged, might be glutamate. Something non-polar might be alanine. Okay. So in order for the protein to do its thing, it needs to have the right amino acids in the right place. So it needs to have the right amino acids in its primary sequence. It needs to have added them at the right point. It needs to have folded them correctly into these alpha helices and beta sheets to get rid of all the water, to have all of that internal hydrogen bonding. It then forms a three-dimensional structure by chemistry between the R groups, so with all of these different types of bonds that hold it all in place. And then if we've got a second protein, we might have some quaternary structure. But that underlying structure, and this is a hideous cartoon, it's quite difficult to show this in 2 dimensions, but all of that internal structure is there to position the right R groups to be on the surface of the protein to then let that protein interact with other things in the cell. So in this case, is a substrate molecule coming in. It could be the membrane, it could be something else. But to get the right amino acids in the right place, to have the right function, you need to have all of this structure right so that R groups are sitting there ready to do their thing inside your protein.